For creating RNA-seq libraries, the VIB nucleomics core uses the Illumina stranded messenger RNA preparation protocol. And you can see the representation of this protocol here on the slide. So you start by purifying total RNA from your samples. And then from that total RNA, you're going to isolate the messenger RNA using oligo-DT primers that will bind to the poly A tails at the ends of your transcripts. Although messenger RNAs are much shorter than genomic DNA, they are still too long for sequencing. So the messenger RNAs are fragmented, cut into pieces using an enzyme, and fragments of a certain size are selected. And then you're going to add random primers, that's the black lines that you see here on the slide. You're going to add random primers to the fragments to initiate first strand copy DNA synthesis in the reverse transcription reaction. So in this way, you are going to create the first strand of the copy DNA. And once you have done that, you're going to create the second strand of the copy DNA. But in this synthesis, you're going to use DUTPs instead of DTTPs, so that uracils are incorporated in the second strand instead of thymines. And in this way, the second strand of the transcript will be labeled with uracils. After that, you're going to add 2 short primers to the ends of your fragments. So the green primer and the blue primer, as you can see here. And these short primers, they can bind to specific sequences on the adapters. So the green primer will bind this adapter and the blue primer will bind this adapter. The adapter sequences are used to initiate PCR amplification, but because the second strand is labeled with uracils, the second strand will not be amplified because the DNA polymerase cannot extend from uracil-containing templates. So it means that in this PCR reaction, you're only going to amplify the first strand of all your transcripts. And that means that you know for every fragment in this library which strand from the transcript it comes from, the first strand. And this also means that all the reads that will be generated from this library, you will know which strand of the messenger RNA they come from. So this is called a stranded library because you have this information for every read, you know which strand of the transcript it represents. If you want to sequence the total RNA instead of the messenger RNA, then you're going to use random primers here in this step instead of the oligo-DT primers. Using stranded libraries greatly improves the analysis of your data. And that's what I try to explain here in this slide. At the top here, you see a part of the human genome, and this is a region where you have a lot of overlapping genes. And that means that you have a gene located on the plus strand, but in the same genomic location, you also have a gene that is located on the minus strand. So they overlap. And this is something that happens a lot in higher eukaryotes. So when you generate unstranded libraries, you will generate reads that will align to this part of the genome, but you will have no clue whether these reads, these sequences, they come from the plus or from the minus strand. So there are 2 solutions here, 2 possibilities. You can either divide these reads equally over the 2 strands, which is not a good solution, or you can ignore these reads, which is also not a good solution. So here at the top, in this part, you see what you would do if you were using an unstranded library. So the reads would map to this genomic location where you have overlapping genes. So they would be divided equally over the 2 strands, and you would get something that looks like this. So with the same number of reads on the plus strand, these are the red reads that you see here, as you have on the minus strand, and these are the blue ones. Yeah, that's the only solution that you can do here. You can equally divide them over the two strands, or you can ignore them, but that would mean that you would lose all these reads that you see here. That's also not something that you want to do. However, when you use a stranded library, and this is what you see here at the bottom, you know for every read which strand it comes from. So you get a distribution of reads that looks like this, and this is the correct distribution, yeah, because here you're sure for every read that it represents the plus strand of your transcript. So you know 100% sure that all these reads, they come from the plus strand, and that all these reads, they come from the minus strand. So this is the actual situation, and you can recreate the actual situation when you have a stranded library. And this is the situation that you would create when you use an unstranded library. And it's very different from what you see at the bottom. So the results here would be really incorrect. So stranded libraries will give much more accurate results. And there are a big plus, especially in higher eukaryotes, where you have a lot of overlapping genes. So we often get questions about the minimum amount of RNA that you need for RNA sequencing. And you can see here the recommendations of the VIB nucleomics score. So with the classic protocol, they assume that you have at least 100 nanograms per sample, but there are specific kits who are a bit more expensive that allow you to go very low. So the last library prep that I want to show is the one for microRNA sequencing. So also here you start from total RNA, but this time what you want is the small RNAs. What's the easiest way to obtain them? Well, by doing a size fractionation. So you put them on a gel, a polyacrylamide gel. And then you just obtain from the gel the smallest fraction. So you get a collection of small RNAs, and typically people focus on these lengths. And here again, you ligate your adapters, you do a reverse transcription to transform it into the more stable copy DNA, and you're ready to go.